All right, hello. In this video, I am outlining muscle contraction, specifically the sliding filament model of how muscles contract. All right, let's start with the structure as usual. And muscle structure is pretty complex and it's a little bit confusing at first, so we'll spend a bit of time on this. You can see we're looking at striated muscle or skeletal muscle here. These are the muscles that are attached to bones via tendons. So the muscles and the bones are connected to each other, and when muscles contract, they pull on bones across joints and allow for movement. And you can see that each muscle is made of a bundle of several muscle fibers, and a muscle fiber is a muscle cell. So that terminology is slightly different than when we were talking about neurons, where in neurons a fiber was referring to an extension of a cell, like either a short extension called a dendrite or a long extension called an axon. Here a fiber, a muscle fiber, is referring to the cell. So a muscle is made of several muscle cells or muscle fibers. And muscle cells are unique in that they contain many nuclei per cell. They're very long cells, they're multinucleate. And within each cell, there are many myofibrils. And so you can see within this cell that they're focusing in on, within a single muscle fiber, there are many myofibrils, each of which is composed of a lot of protein, specifically mostly myosin and actin. And those proteins are arranged in a very specific way in these repeating units called sarcomeres. And when muscle cells contract, the sarcomeres contract. Here we can nicely see how motor neurons interact with muscle cells. So looking at this image, the first thing you should realize is there's two neurons. So they're motor neurons. These are the neurons that take action potentials away from our central nervous system to our muscles. And we're seeing how those neurons are interacting with muscle cells or muscle fibers. And you should realize there are 1, 2, 3, 4, 5, 6, 7, right? 1, 2, 3, 4, 5, 6, 7, yes, 7 muscle fibers or muscle cells shown in this image. So each of those cylinders is a muscle cell, and the little dots within each muscle cell, within each muscle fiber, those little dots are representing the myofibrils, which are those basically the sarcomeres, which contain the actin and the myosin. The motor neurons, you can see they form synapses with each muscle, and that's really important because it's a motor neuron that initiates contraction in muscle cells. And here's a nice zoomed in look at a muscle cell. So again, we can call that a muscle fiber, which is a muscle cell. And you can see there, again, you can see the mitochondria in this one nicely. And you can see the myofibrils, which are those collections of actin and myosin arranged in sarcomeres. And in this one, you can also see nicely what's called the T-tubules and the sarcoplasmic reticulum. Sarcoplasmic reticulum is just the endoplasmic reticulum. It's what we call the endoplasmic reticulum in these muscle cells. You can also see there's a neuromuscular junction there, so you see the axon of a motor neuron. Motor neurons are the neurons that carry action potentials away from our central nervous system to our muscles to initiate some kind of response. And you can see there the synapse between the motor neuron and the muscle cell. Okay, and one more structure, or maybe a couple more structure slides here. Zooming right into the sarcomeres, which are these repeating units that make up the myofibrils. And you can see that a sarcomere is made of myosin and actin. Actin is the orange, or sometimes called the thin filaments, and myosin is the thick filaments, and the myosin has what's called myosin heads attached to the myosin, or as a part of the myosin. So those are sort of like the oars in a rowboat, if you use the rowboat analogy. Other structures that are labeled here, what would you call it? The line that the actin is attached to is called the Z line. And then the part in the middle of the myosin, like the center of the myosin, is called the M line. And so basically, what happens when a sarcomere shortens is the myosin doesn't change size, nor does the actin, but they come closer together. So the space between the Z lines, the distance between the Z lines decreases. And you can see that nicely here. You can see a relaxed muscle on the top where the Z lines are far apart. And then notice as it contracts, they get closer and closer together. And what happens is this dark band where the actin and myosin are overlapping stays the same, but the light band where they're not overlapping gets smaller and smaller and smaller until it eventually, if it's fully contracted, you don't see any light bands anymore, just dark bands, because all the actin and myosin are overlapping. All right, so that's a lot about structure for muscle cells, but I think it's important because muscle cell structure is really complex, and to understand the function, we need to understand the structure. So now getting into how these cells work to facilitate contraction, we're starting with the neuromuscular junction here. And you can see on the top there's the synaptic terminal of a motor neuron. So that's the end of an axon from a neuron coming in. And you can see that it's forming basically like a synapse with the muscle cell. And so in the same way that neurons release neurotransmitters, this motor neuron is releasing a neurotransmitter. It's the same thing. So an action potential reaches the end of the presynaptic neuron. Calcium ions rush into the neuron. Synaptic vesicles move to the membrane. Exocytosis happens, and the neurotransmitter is released. The neurotransmitter for neuromuscular junctions is acetylcholine. So acetylcholine travels across the synaptic cleft. It binds to ligand-gated ion channels in the plasma membrane of the muscle cell, okay, so the muscle fiber, and that's going to cause sodium ions to rush in and initiate an action potential in the muscle cell. Okay, so an action potential happens in the muscle cell. That action potential is propagated down into the cell, like into the inside of the cell, by way of these T-tubules. So the action potential is propagated along the T-tubules until it reaches the sarcoplasmic reticulum, and the action potential causes the sarcoplasmic reticulum to release calcium ions. So that's how we go from an action potential in a neuron to the first stages of contraction in a muscle cell. So just zooming in a little bit on this image here, the orange protein is the actin, and the purple one with the oars on it with the myosin heads, the thick one, is the myosin. So we have the thin actin and we have the thick myosin. And on the actin there are binding sites for those myosin heads. Those binding sites are normally blocked by a protein called tropomyosin. And so the tropomyosin is sort of the long string-like protein that's going around the actin. And occasionally there are these sort of purple protein complexes. Those are called troponin. So the purple proteins on the actin, it's getting complicated, are troponin. And the sort of threads attached to the troponin are tropomyosin. So what happens is when the calcium ions are released from the sarcoplasmic reticulum, they bind to the troponin, and the troponin shifts over, which then causes the tropomyosin to move, because they're all connected, and that exposes the binding sites on the actin for the myosin heads. So now muscle contraction can occur. Without that step, muscle contraction can't happen because the binding sites for the myosin heads, which are on the actin, are blocked by the tropomyosin. And here you can see that nicely shown in the zoomed in look here at the actin filament with the myosin binding sites. And on the top, you can see all those binding sites. They're sort of represented by little holes inside the actin subunits. Those binding sites are blocked by the tropomyosin. And you can see how the calcium ions, represented by the little green dots, they bind to the troponin, to this troponin complex. It's more than one protein. And that causes the whole thing to shift over and exposes the myosin binding sites. So the myosin heads can now bind to those sites on the actin. And then contraction can happen. So this is really where we get into this sliding filament model and all the steps involved. So let's go through it sort of one step at a time. And remember, this is after the binding sites have been exposed. So calcium has been released, binding sites are exposed. Now we can get contraction to happen. And so initially, you can imagine ATP bound to a myosin head, but it's still ATP. And the myosin head is in its low energy configuration. It's not bound to the actin yet. Then the ATP is hydrolyzed into ADP, an inorganic phosphate. That energy, so that releases energy. Anytime we hydrolyze ATP, we release energy. That energy is used to change the conformation of the myosin head to its high energy configuration. Then a cross bridge forms between the myosin head and the actin filament. So the myosin head can bind now to those myosin binding sites on the actin filament, forming a cross bridge. Then the ADP and inorganic phosphate are released. Now, depending on the source you look at, this might be described in a more detailed way with the phosphate being released first at a different time. Don't worry about that level of detail. You just need to know that the ADP and inorganic phosphate are released at some point, and that causes the myosin head to return to the low energy conformation, which is basically causing it to pull inwards. And that's pulling the actin filament towards the center of the sarcomere, and it's shortening the length of our light bands in our sarcomere. And then you can see ATP comes in again and binds to the myosin head. That's what causes the myosin head to release. So the ATP binding causes the myosin head to release. ATP hydrolyzing causes it to change to its high energy configuration. And ADP and inorganic phosphate releasing causes it to shift to the low energy configuration. And that just cycles through repeatedly until the binding sites are blocked again, allowing for muscle contraction. Just one more little detail to add to this is the detail related to titin. And so here you can see I am in the middle of a sarcomere. I'm at the M line, right? And those thick, in this diagram, the thick green filaments, I'm on top of one, are myosin, and the thinner green, sort of, they look like two spirals, those are the actin. And in this diagram, the orange things, the orange spring-like structures, those are titin. So titin proteins are very long proteins, and so they connect from the, you can see here, from the myosin, from the myosin to the Z line. So you can see these titin fibers connected there. And when the muscle is relaxed, you can imagine those are being stretched. Those are extending. Can you see that? Because the Z lines would move further apart when the muscle is relaxed. And those would then extend, and they would, just like a spring, if you pull a spring apart, it's storing energy now. It wants to return to its original shape. So when muscle is relaxed, the titin molecules store potential energy, making the next muscle contraction easier. And then when muscles contract, those titin molecules release that potential energy and therefore help facilitate that contraction. They make it easier so the muscles can exert more force. So that's an extra detail about the role of the titin fibers in storing and releasing potential energy to increase the amount of force that muscles can exert. And that is the end of this presentation. I hope you found it helpful. Thank you.